All right. And with that, we're really excited to introduce Dr. Kristen Aquilino today, who is the lead scientist for the white abalone captive breeding program at the University of California, Davis's Bodega Marine Laboratory. Her research focuses on the reproductive conditioning and husbandry of endangered white abalone, as well as the effects of climate change and disease on their captive and wild populations. She's a member of the California Sea Grant Extension Team, connecting her work with state and federal agencies, indigenous nations, commercial aquaculture, growers, aquariums, and other stakeholders. And finally, Dr. Aquilino is a recipient of NOAA's Species in the Spotlight Hero Award. So we're really excited today. We'll let you take it away, talk about the work you do with saving white abalone. And I'm going to stop sharing now.
All right, awesome. Thank you so much, Maggie. I'll just start by thanking everyone for joining this. I'm really excited to talk about white abalone. As Maggie said, I kind of wear a few hats in this. I'm the lead scientist for the white abalone captive breeding program, but I also do outreach and extension for California Sea Grant, and I work really closely with NOAA. So even though I'm based at UC Davis Bodega Marine Laboratory, many of our programs are completely funded by NOAA, and our collaborators and recovery coordinators are at NOAA. So I'm just going to start with a question for everyone. And Maggie said I could use this poll thing. So I'm going to ask that you raise your hand if you've ever eaten an abalone. So up at the top left, there's a little, looks like a little person with their hand raised. And if you click on the arrow and drop down, you can raise your hand. And I'm curious to know if anyone else has eaten an abalone.
Sorry, Dave Bader had raised his hand. I kind of unraised his hand, but you can also type it in the chat box if you can't raise your hand on Adobe.
Yes. I'm curious. Okay, so we have at least 3 of you who have eaten abalone. Has anyone ever dove for abalone? You can unraise your hand if you've eaten them and not dove for them. So we have like 1 person maybe who's dove for abalone. So depending on the groups I'm talking to, many people who are not on the west coast of North America have never even heard of abalone. A little bit of my background, I'm from Iowa, which is about as far from the ocean as you can get in the United States. And I didn't know what an abalone was until my mid-20s, and now I've received a national award for trying to save them. So we can all learn quickly here. And I think it's really interesting that abalone are kind of like lobster to Maine or blue crabs to Chesapeake, but there tends to be more of a local knowledge and history of them, that people throughout North America don't tend to have interacted with these animals as much as some of the other iconic species in the US. So I think that's really interesting. So I'm just going to start, because some of you might have less experience with abalone. I'm going to share my screen now, and I'm going to start by talking a little bit about white abalone and what are abalone in general. So hopefully you can all see my presentation now. While I'm in my presenter view, I can't see the comments, and I also can't see my own face. So I think you can still see my own face. And if you have comments or questions, please feel free to still put them in the comment box, and hopefully Maggie can relay those to me via audio. Maggie, is that good?
Yes. Yeah, that's perfect.
Cool. So what's an abalone? On the left here, you can see a picture of what a lot of people think of as an abalone. It looks a lot like a rock, especially when it's in the ocean. It's covered with sponges and worms and boring clams and all sorts of things that make it look a lot like a Muppet. But in reality, it's a snail. It has this face. It's really quite cute. I think it has 2 beady eyes in the front. Its mouth is in the center. It has these long tentacles that we call cephalic tentacles, and then it has little tentacles all along the side that we call epipodium. And it has gills that are underneath those holes on top of its shell, the holes that we call the respiratory pores. So the gills are here, those respiratory pores are here. White abalone is probably called a white abalone because the tissue underneath those pores is very white. So even if you saw this thing in the ocean that looked a lot like a rock, you couldn't tell it was a snail, you might still see these 4 white dots, and that's how you might identify it as a white abalone. And white abalone is one of 7 species in the spotlight in NOAA, the species that NOAA deems most likely to go extinct in the very near future without our help. Those other species include the leatherback turtle, beluga whale, southern resident killer whale, Hawaiian monk seal, and a few salmon species. And I like to joke a little bit that some of these species may at first appear a little more charismatic than the white abalone, but remember this picture here, that they do have a face. And I think that part of our job as aquarists, as scientists, as people interested in marine conservation is to try to get people to connect to these species, to do our work to get their numbers up, but also to get people to care about them. And we care about things with eyes. So this is definitely my favorite picture of a white abalone for that reason. And abalone have a long distribution up north and south in the United States, and there are 7 species on the west coast of the US. There are 56 species or so throughout the world. They occur on every continent except for Antarctica. And these colored lines here indicate the north to south distribution of the 7 species in North America: the pinto, flat, red, black, green, pink, and white. Of those, whites and blacks are listed in the ESA as endangered, and pintos are listed as a species of concern. But all of these species have been fished, and many of them heavily depleted, either locally or throughout their range in commercial and recreational fisheries. But we really care about them. So I kind of mentioned that people outside of the west coast of North America often haven't heard of abalone in the United States. And so why is that? Even though they're so important here, so culturally, people have been harvesting these animals for food and for jewelry and even currency. Some native nations used abalone shells as currency for thousands of years. So the first black and white picture here is of a shell that was collected from a Native American midden on Santa Cruz Island and is estimated to be over 7,000 years old. These animals are really, really important to the traditions and cultural history and identities of Native people here on the west coast of North America. And there was, until very recently, a very popular recreational fishery for red abalone on the north coast of California. So this is a picture of me diving in Fort Ross Cove a number of years ago. And lots of people were doing that. It was bringing $45 million a year into the state of California. It was a really important fishery. And with the closure of that fishery in 2018, due to a variety of environmental factors that really depleted the population, it's been a really difficult economic situation for many people on the north coast of California. Lots of businesses shutting down. That economic impact spills into aquaculture. So there's a number of important aquaculture farms in California for red abalone. This is a picture of Doug Bush, who manages the cultured abalone farm and the stack of market-sized abalone, which also bring millions of dollars into the state. It's a really sustainable way of producing protein. Abalone eat kelp. Kelp grows really fast, and these facilities are really dedicated to growing protein in a sustainable way. Another picture here is of another abalone farm that recently shut down in California called the Abalone Farm, a creative name. And they also had a really important product for the state. Their ecological importance is also notable. I like to call them the Zambonis of the seafloor because they maintain this really beautiful color of cover of crustose coral and algae. So this is a white abalone in the wild, and this pink stuff around it is a species of encrusting algae called coralline algae, which serves as a landing pad for abalone and a lot of other marine invertebrates to settle. So it's a really important part of the habitat. And without that, these animals, many of which are really important for preserving the kelp forest ecosystem, that without it, it doesn't exist. They don't have a place to settle. And abalone do compete with urchins. So we have situations where we don't have healthy abalone populations. We often have too many urchins in these urchin blooms. It's not something that we have a problem with in Northern California right now, with far too many urchins in the habitat, which is hard to recover from. So these cultural, economic, and ecological importances of abalone in general, these are themes that will continue throughout my talk. In terms of the decline of U.S. abalone species, it was mostly due to overharvest. And I want to be very clear here that when we talk about overfishing, it's important to acknowledge that, at least for the case of abalone, this isn't true for all fisheries, the fishers were following regulations. There is poaching for abalone, and it is significant, but the overharvest really had to do with poor management of the fishery. Not anyone's fault. We didn't really understand the ecology of the species and the population biology of the species until it was too late for some of them. Abalone commercial harvest started in the 1850s, mostly with Chinese immigrants, mostly with rock picking. Japanese immigrants brought diving technology to the fishery. And as that technology got better and better, we were able to stay down longer, take more and more abalone, take deeper abalone than we were before. And there was, in the 1940s and 30s and 40s, there was a song that basically went, the more we take, the more they make. We could never deplete the abalone. We really thought that because they're broadcast spawners and sending millions of gametes into the ocean, we wouldn't be able to deplete them. But we took a lot of abalone. And so this is a picture of an abalone midden, or I guess an abalone mound. This wasn't a native midden, an abalone mound of shells from Santa Barbara from the 1940s or so. And this was a really common sight through the open period of the fishery from the 1940s until through the 1960s, 1970s or so. Another challenge that abalone have had in the wild in California is something called withering syndrome. And though this wasn't the main cause of the depletion of most of the abalone species in California, it hit black abalone really hard. And it's an interesting disease because it's caused by a bacterium that affects the esophagus of the abalone. The bacterium is called Californiensis Candidatus xenocaliforniensis, CAXC for short. And that bacterium can affect the esophagus of the abalone. In cold water, the abalone are apparently asymptomatic. And then when the abalone are in warm water, the bacteria proliferate, the abalone stop eating, and instead they digest their foot muscle and fall off the rock and die. So on the left here is a healthy red abalone. This is its foot, its shell's on the back side. Its foot covers its entire shell. You can hardly see the shell margin on the right side here. On the right is a diseased abalone. This abalone has withering syndrome. Its foot is very shrunken. It has digested that foot muscle because it has stopped eating. And I mentioned this really impacted black abalone. Black abalone are the shallowest of all of the species on the west coast of North America. There was an El Nino period in the 1980s. That was the first time that we noticed the effects of this disease, and it really decimated a lot of black abalone populations, and it's one of the main reasons that they were listed in 2008. So what about white abalone? White abalone is the deepest of all of the species on the west coast of North America. The remnant population is mostly below 100 feet or deeper. We think that historically they were as shallow as 30 or 40 feet, but they tend to be deeper water species, and it's probably why they were one of the last to be fished in the fishery. They weren't fished heavily until the 1970s or so. But in about 10 years, in the 1970s, we harvested about 99% of white abalone in the wild. So we estimate that there were about 280 or more metric tons of white abalone fished, and there are fewer than 2 metric tons remaining. Only a few thousand white abalone remain in the wild. And this is very problematic for a species like abalone because they're really terrible at long distance relationships. And most of the abalone that are left in the wild are so far apart from one another that as broadcast spawners, their gametes will never find each other. So in some ways, we could say that this population is already effectively extinct, even though there are thousands of animals left. They are really on the brink, and it's very important that we act quickly to try to enhance the captive population, or the wild population. They were first federally listed as endangered in 2001. And at the time, we thought they'd be a relatively easy species to save. Not only are there abalone on every continent except for Antarctica, there are also abalone farms on every continent except for Antarctica. We know how to make lots and lots of abalone. So the plan was to take some white abalone from the wild, some of the few that were left, into captivity, spawn them, produce hundreds of thousands or more babies, and put those babies back out into the wild. So step 1, captive production. Step 2, outplanting into the wild. And things kind of started that way. So in the year 2000, they collected about 20 wild white abalone and brought them to a facility in Southern California called the Channel Islands Marine Research Institute. And they spawned those animals in 2001 and created over 100,000 juveniles. So here's Tom McCormick, who is leading the program, and he had this great production really early on in the program. So compared to the few thousand left in the wild, this was looking really good. Unfortunately, when those animals were just about a year old, 95% of them died from disease. That disease is withering syndrome, the disease I mentioned earlier. And at the time in 2001, we had just discovered what that disease was. So even though we had seen those black abalone dying in the 80s, we didn't really understand why that was happening until the late 90s. And there was nothing that we could do about it. And Tom was really confined because he was restricted to using this warm seawater that was coming in through a power plant, and it was very difficult to try to figure out how to deal with this disease. So NOAA looked for a place that could house the white abalone captive breeding program where animals might do better and that might have the expertise to deal with this disease. And they looked no further than us here at Bodega Marine Laboratory. We have a lot of expertise here in the development and reproduction of invertebrates, ecology and outplanting of abalone, and in shellfish pathology. So back up just a little bit here is this disease was first discovered in the Channel Islands. It has slowly moved its way north. And in fact, when Bodega Marine Lab first got the permit for this white abalone captive breeding program in 2011, the bacterium hadn't quite made it up this far yet. It did very shortly thereafter. So it is here now. But this pathology research has been really, really important. Particularly, the state's Shellfish Health Laboratory, California Department of Fish and Wildlife's Shellfish Health Laboratory, is located here at Bodega Marine Laboratory and is headed by Jim Moore. And he and his colleagues have been really instrumental in identifying and tracking and learning to treat this disease. So they actually developed an antibiotic bath treatment that you can put the animals in. It rids them of the bacterium, and then as long as you put them on treated seawater, they are healthy. And that's been a really key part of the success of this program since moving to Bodega Marine Lab. And as an aside, I also say that Jim and his colleagues have developed a shell waxing treatment. I mentioned that these white abalone that come in from the wild often look like Muppets with everything growing on their shells. Those things growing on their shells can bore into the shells, they can open up space for infection, and they can also make the shells very brittle, which we don't want when we're handling these animals. So this waxing treatment is a combination of organic beeswax and organic coconut oil, and we paint it on the shells with a paintbrush, as you can see here, and it basically suffocates any of those boring organisms and makes the shells healthy. So that's a fun thing that we get to do twice a year. We get to do spot treatments for all of our abalone. And I'd like to say that between the cleansing antibiotic baths and the exfoliating waxing treatments, we have the most pampered snails in the world at Bodega Marine Laboratory. Though the program is centered here and the permit is held by UC Davis, this is still a very large collaboration of partners all over California and Mexico as well. The range of white abalone goes down into Baja California, Mexico, and for a listed species, we as the United States of America are required to restore it throughout its range before delisting it or downlisting it. So this is a smattering of many of the partners in the captive breeding program. I mentioned NOAA is our main funder and a big part of this program. California Department of Fish and Wildlife is also a major part of this program. And then we actually hold white abalone 11 different facilities. Just like you don't want all of your eggs in one basket, you don't want all of your animals in one tank on the San Andreas Fault as we are here at BML. So we have animals at many different facilities, and many of these facilities are able to bring different skill sets to this program. So we have lots of academic institutions and research institutions that bring genetics expertise or other types of things that we don't necessarily have as much at UC Davis Bodega Marine Lab. But what's extra special, I think, is that a number of these are public aquariums. So we have animals at California Science Center, Cabrera Marine Aquarium, we have them at Aquarium of the Pacific, and at the Santa Barbara Museum of Natural History Sea Center. So these are places where anyone can go and see these animals. And that's really, really important for saving the species, is getting people to identify with them, as I mentioned earlier. We also have farms that we collaborate closely with, and this is important just for sustainable aquaculture. We trade a lot of information with the farms, and basically we take their best methods and then tweak them for white abalone to try to make them even better. And then we do science that helps them figure out how to run their farms more sustainably and more efficiently, especially in the face of climate change. And we've had a lot of success. So when I first started with this program as a postdoc in 2011, we just had a handful of adult white abalone. Our first successful spawning season was in 2012, and we created 20 adult white abalone. So this graph has the number of year-old white abalone produced annually and the year they were spawned, basically. So there's those 20 in 2012. We've increased really steadily, and we're about to count the animals that we produced in 2019 next week, and I predict that we have 30,000 or more. from this year. So this is awesome. This is a great amount of success, considering that in about the 10 years or so before 2012, there was no captive breeding at all, successful captive breeding of white abalone, a lot because of the challenges dealing with disease. So this is awesome, but this 30,000, even if we sustain it, is nowhere near that 100,000 that they were able to produce at Simri during that first year. So much of my work is trying to do the science to figure out how to bring us back up to that number, because in order to save the species, I think we do need to be putting out 100,000 or so animals every year in concentrated areas, so that way they can survive and reproduce on their own in the wild. So in order to tell you a little bit more about our science and our research, I first want to show you a video of their development. So this is the video here, and there should be no sound. So don't think you're going to hear, well, you should hear me, I guess, when we're playing this video. And this will give you a little bit of sense of how do we do the captive breeding, what methods do we use, and what is the development process for these animals look like. So this is at UC Santa Barbara. When we spawn these animals, we check their gonads. It's a really romantic process. We put them each in their own bucket of hydrogen peroxide, a really romantic love potion that gets them to give us whatever gametes they have. So I think Emma Horanek is on this call, and she helped with a number of these spawning attempts with the animals at UCSB. Males release sperm, females release eggs, and we mix those eggs and sperm together at a really precise ratio to prevent polyspermy, because abalone can allow more than one sperm to enter each egg. So we want to make sure that that doesn't happen, because then development won't happen. Here's a fertilized abalone egg, and each of those little ticks around the side is a sperm. That abalone cleaves after a few hours and develops into an embryo. This embryo stays at the bottom of the ocean or the bottom of a tank for about 24 hours and hatches out into a swimming larvae about 24 hours after fertilization. It has a ring of cilia around itself that it uses to swim as it develops. And to the naked eye, it looks a lot like dust or little vanilla bean seeds. You can hardly see them, but you can tell there's something there with the naked eye. They start to develop their shell when they're just a couple days old, and they also develop an operculum on the front growing edge of their shell so they can close their soft parts in. They have a yolk from their mom during this time, so they don't eat at all. They completely rely on this yolk from mom in order to develop. We teach them to do somersaults. They're like little bumper cars when they're on a slide. And we kind of make sure they're developing as they should. We do regular water changes at this time. Once they start developing eye spots, which you'll be able to see even better in the next picture here, here's their eye spots, and they start to develop their cephalic tentacles, they tend to spend more time on the bottom of our larval culture chambers. We know at that point they're about ready to settle. To settle them, we use a solution called GABA, which gets them to relax and go to the bottom. And they metamorphose. They lose their cilia, and they transport themselves from something that swims and doesn't eat to something that crawls and eats for the first time. And this is a really energetically costly process to go from such different life strategies, kind of like a caterpillar becoming a butterfly. And we expect 95 to 99% mortality just during the first 3 months of life because of this transition. Once they do settle, they look a lot more of what we think about when we think of an abalone. They start to develop their shell. Their shell at first is transparent, which is cool because you can see their guts inside. You can tell they're full of diatoms. This abalone is about to poop right now. There, that's the cutest poop you've ever seen. When they get a little bit older, they start to develop their respiratory pores, the holes on the top of their shell. So this animal has just started to develop its first respiratory pore. So I'm going to stop the video there and go back to my slideshow here. So that this is some background in order to see what that life cycle is like. And now I'm going to talk a little bit more about the research we're doing to try to increase production with that background in your head. The first is try to figure out what to do about that post-settlement survival. So I mentioned 99% of them are not going to make it till 3 months old. And that's huge mortality. So, and this is true not just for white abalone, but for red abalone aquaculture, whose methods are down pat. It's kind of the strategy of broadcast spawning organisms. You produce a lot of gametes really cheaply, and you don't expect a lot of them to make it. But when we're dealing with millions of larvae, increasing survival through the post-settlement stage by even 1% is a heck of a lot of animals. So that's a really important touch point for increasing production of white abalone. The next part is reproductive conditioning. I mentioned the really romantic love potion that we give them to give us whatever gametes they have. They're usually giving us way fewer than we expect they should. And so what can we do to get them to become more reproductive? Their genetic integrity, we have very few brood stock because there's very few animals remaining in the wild. So how can we make sure we're managing their genetics as best as we can? And then how can we prepare these animals for climate change? The animals that we are putting back out into the wild are going into a very different ocean than their grandparents experienced. And we want to make sure that they do okay there. We also want to make sure that our production on the land side of things can keep up with climate change or mitigate the effects of climate change. So I'm going to go through each of these 4 things in a little bit more detail. In terms of post-settlement survival, as I mentioned, that's a really important touch point. We could make a big change given the numbers of animals that we have at that point. All the methods that we're using so far are really based on those aquaculture methods that abalone farms are using. But there might be things like, what if we use a slightly different concentration of GABA to settle them? What if we use a more natural settlement substrate like crusting coral and rock? What if we give them different food as their first food? Right now we're using a species called Navicula, which is really common in aquaculture, but maybe they need more of a variety. Maybe they need a different species. And then also, what can we do during their larval phase to make sure that they are more equipped to get through settlement? They only have that yolk from mom to get them through that entire first week period, all the way through metamorphosis. So maybe we can do a better job of provisioning mom so she can provision her eggs better, and therefore her babies can make it through that period even better. So those are the kinds of things we're thinking about with post-settlement survival. And already, I think much of our success that we've had, that increase in success over the past few years, has to do with tweaking how we're doing the settlement process, with really getting the right kinds of foods for babies, with really provisioning mothers better, those kinds of things. So we're looking forward to exploring some of those things more. The next thing is getting them to produce more. So we put them in this hydrogen peroxide solution. They give us what they have. Usually females are giving us on average about 600,000 eggs a piece for the first 5 years or so that this program was at Bodega Marine Lab. And they should be spawning millions of eggs a piece. So what are they missing? Often males aren't spawning at all. There's a video here and a few pictures of males and females spawning. This number of eggs here is probably around 300,000 or so. We would expect many, many more from a female. This male is, the amount of sperm in this bucket is typical for a male. We try to collect it right when it comes out of the respiratory pore, so it's as concentrated as possible and it works the best as possible. But if a male really is spawning well, this bucket should be opaque with sperm. We should not be able to see the abalone in it anymore. So I think that even though this induction method is working well, telling them to give us what they have, there's probably some important environmental cues that let them know in the wild that spring is coming. They should invest in their gonad. White abalone tend to reproduce mostly in the late winter to early springtime. And in the wild, they might have some indication that that's the time of year. In captivity, on regular light cycles and constant temperature and filtered seawater, maybe they're missing some of those cues. So we have looked at a number of different ways to reproduce or to get our animals to be more productive. The most basic and easiest is diet. Abalone love kelp. They love macrocystis. It's like a Twinkie to them. They will jump on their friend's back in order to reach kelp. It is really delicious. They eat it quickly, and it's good because there's a lot of calories, but it doesn't have a lot of nutrients. So we have a tumble culture of dulse. We also use other types of red algae that tend to be much more proteinaceous in order to try to condition our broodstock so that they have more investment in gametogenesis. I mentioned some of those other things. Besides diet, we're thinking about temperature. So this is a picture of some of my technicians. We are looking at different temperature regimes to see which ones might better induce gametogenesis. We're thinking about photoperiod. So our lights in our lab are all attached to an astronomical clock that go on and off the same time the sun rises and sets in Santa Barbara, which is the center of their native range, because maybe that might help them become more reproductive. We're starting to look into hormones to see if we might be able to predict when gametogenesis is happening. We might be able to induce gametogenesis, and we might even be able to induce spawning by using hormones such as GnRH and a few others. We're also thinking about disease and how that might affect reproduction. Jim Moore and his colleagues, the shellfish health pathologist and his colleagues, have done a really great job of understanding what are lethal conditions for this disease. So we know that if a white abalone has CELC, has the bacterium that causes withering syndrome, and is in seawater that's 18 degrees Celsius or more for a period of days, then it will succumb to this disease. We have a really good sense of that. But maybe there are temperatures or conditions that manifest other symptoms in the abalone, and we would like to know what those are. So maybe at 16 degrees Celsius, they stop becoming reproductive or less. And so this experiment actually with the technicians in this picture here is crossing both temperature and disease to see the effects of each of those on white abalone reproduction, and that's funded by California Sea Grant. And then on top of that, we're looking at climate change and how that might affect reproductive condition, because the multiple stressors of some of these things might add up to different effects in the abalone. And there's a picture here. Female is spelled wrong with a C at the end, but this is a female white abalone. Its gonad is kind of a grayish color. Those are eggs right there. And this is a male white abalone. Its sperm is a little bit more yellow. And one really cool thing about working with white abalone is they become reproductive at a very young age. So these animals are only 14 months old, which is very young for a lot of abalone species to be exhibiting gametogenesis. And even though we're not sure how viable eggs are at this age, to do these studies, we have a lot of animals available. And so the first few years we were doing this work, we really wanted to know what was driving gametogenesis, but we just didn't have enough animals to ask these questions. And now we are in a place where we can do that, which is really exciting. And I'll talk a little bit more about climate change when I get to that section as well. So far, these methods seem to be working somewhat. We've had a lot more success in spawning in the past few years than we did the few years before that, but we really are just getting into trying to figure this stuff out more experimentally, as opposed to just plugging in what we think are the best conditions and crossing our fingers and hoping that they work. Genetic integrity of our broodstock is really important. For the first few years that I managed this program from 2011 until 2016 or so, we only had one wild-caught white abalone among our broodstock. We had about 40 or so, depending on the year, captive-bred white abalone broodstock from a 2001 spawning and a 2003 spawning that represented only 3 total parents. So genetically, we only had 4 total individuals, and that captive male never spawned in captivity and still has not. So we were really limited in terms of genetics. And even though the main thing is to get numbers of abalone out there, we also want to somewhat represent the genetic diversity that was out there, or at least is currently left in the population, especially because we know these stresses like climate change might need to act on more variation than currently exists in the captive population. So we were lucky to get a permit from NOAA in 2016 in order to collect new wild white abalone broodstock. This is one of the first ones that came in from the wild here. And we have 10 of those animals now. And we have folks that we collaborate with at NOAA Southwest Fisheries Science Center in La Jolla, like John Hyde, who are helping us be able to determine what are the best crosses that we can make, what are the best ways that we can manage the genetics that we have. But we're also thinking about things like sperm cryopreservation to be able to have more control over which crosses we're making. So right now we have often very few animals spawning. Sometimes spawning attempts are not successful because only females spawn or only males spawn. If we're able to cryopreserve sperm, that means that those situations suddenly become successes. If only males spawn, we can cryopreserve those sperm. So then when only females spawn, we are able to fertilize those eggs. And also we are able to make crosses from animals across institutions, which we wouldn't necessarily be able to do as easily otherwise. So we're just starting to develop those methods. And we are in a lot of communication with folks at the Puget Sound Restoration Fund that are trying to develop these methods for pinto abalone. and people at CELC and Ensenada who are also developing these methods for other species of abalone, because it seems like they're very, very different techniques among the different abalone species. And then the other thing that I'm really involved in is trying to figure out how climate change may impact both the captive production of white abalone and what happens to them when they get in the wild. My closest collaborators with this are Sarah Bowles, who's a PhD student here at UC Davis, and she's just finishing her dissertation. She's a geneticist, and she's thinking about the genomics of climate change. And then Dr. Dan Sweezy, who's the lead scientist for the Cultured Abalone Farm, and has a joint appointment here at UC Davis Bodega Marine Laboratory. I also work closely with the Bodega Ocean Acidification Group at UC Davis Bodega Marine Lab, which includes Brian Gaylord, Tessa Hill, Eric Sanford, and some other colleagues. So we're thinking about what levers can growers, whether you're doing white abalone restoration, pinto abalone restoration, or commercial aquaculture for abalone, what levers can growers pull in order to mitigate the effects of climate change, particularly ocean acidification? So we're thinking about seawater buffering at larval stages and other life history stages, maternal diet, post-settlement substrate and diet, temperature, photoperiod should not be on there. Sorry, that was left over from before. And then disease as well, because all of these things might change how captive production is happening and how captive or wild white abalone are responding to it. And so far we know that, or we've been able to tell that white abalone, even if we don't buffer seawater during the larval phase, we still see pretty good survival through the larval stage, but many of them kind of poop out after that. And so we do think that they are seeing, there is an effect of ocean acidification on these animals, especially when they're exposed at a very early life history stage, and especially for the species that requires only eating yolk from its mom. Basically, it gets all of its nutrients from that yolk. It can't compensate by changing its metabolism during its larval phase in order to survive later on and get through that metamorphosis. And then there are a number of other teams thinking about field research and outplanting. So Cal Fish and Wildlife, the Bay Foundation, Powell Marine Research Group, and NOAA have been really trying to think about what's the best way to put these animals out in the wild. They've done a bunch of pilot work with red abalone. California Department of Fish and Wildlife has mostly been using these baby abalone recruitment traps, or BARTs, which are basically lobster traps that are modified and filled with broken up cinder blocks and create really nice interstitial habitat for abalone that are small and need to avoid predation. And then the other enclosure that's been tested is a short-term abalone fixed enclosure, or a SAFE, which is an enclosure that abalone cannot actually escape for the first few weeks. So abalone are put in this enclosure, they're fed kelp, and the idea here is to hopefully dissuade some predation. When you put a bunch of bite-sized abalone in the ocean, it attracts a lot of predators. It's like ringing a giant dinner bell for them. And so perhaps by putting them in the SAFE, the predator queue either becomes less over time, or the predators become desensitized to it, and the SAFE is then cracked open after a period of a few weeks, and the abalone can crawl out. So we are testing both. We have tested both these methods in tandem, or these groups have, and they are both also being tested with our very first release of captive white abalone ever. So this November, we put white abalone in the ocean, for the captive-bred white abalone in the ocean for the very first time, which was really, really exciting. I got to be there for the first time we put them in the ocean, and I sat on the deck of the boat and I cried seeing my babies go out in the ocean for the first time. There's definitely still some out there. This video was taken last week during a survey. There's 12 of the baby white abalone that were bred at UC Davis Bodega Marine Lab on this tile here. If you want to read more about this, there's a really great LA Times article that came out after the first outplanting, and you should definitely look it up. There's a nice video there as well, where you can see the divers putting the animals in the ocean for the first time. So I have a few take-homes, and then I will open it up for questions. The white abalone are really culturally, economically, and ecologically important. We do care about these animals, and they do matter to us. And they will go extinct very soon without our help. We have the tools to save them, and they should be a relatively easy species to save. Their habitat is relatively intact. So as long as we can make enough of them and make them genetically diverse enough, then it's not like we're trying to save a species where there's very little habitat left for them. And the tools we're using to help save white abalone will really enhance the sustainability and security of abalone fisheries and aquaculture elsewhere. There are a lot of other depleted abalone species, and abalone are pretty tasty. So there's a lot of ways that our work is interacting with a lot of other important needs. So I will open up for questions in a minute, but if you did not get your fill of white abalone today, we are on social media. I'm on social media, Facebook, Twitter, Instagram, and white abalone just got a TikTok account recently. My email address is here. If you're ever in the North Bay area and are interested in seeing our facility, I love to give tours of our lab. I force people to take shelfies with me, whether you're Janet Napolitano or Kathy Ann Sullivan, the first American woman to walk in space, or our UC Davis chancellor. I really enjoy giving tours, so if you're ever in this area, I'd be happy to do that. And so I'm going to stop sharing my screen, if I can figure out how to do that. Let's see.
Thank you so much, Kristen. That was a really great presentation. I definitely learned a lot about abalone.
Yeah,
so if anyone has any, feel free to type them in the chat box or just speak up. You can unmute your speakers and. Give people a second to think about it. Do you have a question?
Hi, this is Christos Mikalopoulos with NOAA. Are there any species of abalone that are still harvested commercially?
None in California, and actually none in the United States at all. I don't think Canada has any either. Most of the species in Baja are still harvested commercially in Baja, California, Mexico.
How about recreational harvest?
There's also no more recreational harvest. So the closure of the red abalone fishery north of the Golden Gate in 2018, that was the last remaining recreational fishery for abalone in the United States.
Was there like outcry about that, or did people mostly understand that they were almost extinct, and was there a fight?
I mean, hopefully red abalone are not almost extinct. Hopefully they will be coming back. There was definitely an outcry about it, but the environmental impact going on in the north coast of California is really unprecedented. So Cal Fish and Wildlife are calling it the perfect storm. There was a harmful algal bloom in 2011, followed by sea star wasting disease, which is a really important predator of urchins. Sea stars are important predators of urchins. And as there was a really huge urchin recruitment with the decline of the sea stars, there was also the warm water blob followed by an El Nino, which decimated the kelp in Northern California. So for 2 or 3 years, we had a 95% or more loss of kelp in the Sonoma and Mendocino coasts. And because of that simultaneous urchin bloom, the kelp has not been able to recover. So oceanic conditions have returned. We have cool seawater again. The kelp should be able to grow fine, but there are so many urchins in a very, very large stretch of area that the kelp hasn't been able to come back. And there have been urchin barrens before in many different places. Most of the time, they're much smaller scale geographically than what we're experiencing on the north coast of California right now. And so people are trying to figure out what to do about this. There's a group called Kelper that's thinking about trying to do kelp restoration. There's a group called Urchinomics, which is trying to figure out if there's a way to ranch urchins. Basically, one of the challenges with the urchins that are out there right now is that they have no commercial value because they're starving too. So they don't have a gonad, they don't have uni. And so are there ways to make those marketable in some fashion? But yeah, it's a really terrible environmental disaster. And it is sad. I mean, I didn't mention some of my connection with abalone. I met my husband surveying red abalone with Cal Fish and Wildlife on the North Coast. Our second date was diving for abalone recreationally. My engagement ring, which I still wear years after marriage, is an abalone pearl. We couldn't wait to teach our kids someday to dive for abalone, and it is devastating that we might not be able to do that now. And I'm hopeful that some of the things that we're doing with white abalone will help inform the recovery of the red abalone on the North Coast.
Thank you. We have a couple questions on the chat. Slady2 says that, I know octopuses are the villains for abalone. How much of the recently released abalone do you expect to survive till they are large enough to escape octopus predation?
Octopus are tough. Yeah, you can't keep them out of anywhere, and you often can't tell they're there until you can tell they're there. We are expecting very few of these animals to survive the outplanting, and we actually feel like from other species, work with other species, we probably won't know for a few years what the success of these first outplantings is like. So with pinto abalone in Washington, they're about 7 years ahead of us, and what they're seeing is they put a bunch of pinto abalone out in Washington. They don't see any for 2 or 3 years, and then suddenly they see a bunch of them. And I think this is because the ones that are surviving are not only cryptic to all of those predators like octopus, they're also cryptic to the divers. And it isn't until they're large enough to escape that predation that they become emergent and they become visible to divers. So this is going to be a challenge in terms of continued funding and continued effort, because we might not know what's going on for a while. The good news is that we have a lot of pilot data from red abalone that we can use for this, and from the pinto abalone work. So we're tracking the number of shells that we're seeing. And right now, the number of shells that we're seeing from our first white abalone outplants are pretty similar to the ones that we've seen for red abalone. And I want to give another caveat about lobster. Lobster is also a really big predator of abalone in Southern California. And we initially thought we might put these abalone out in marine protected areas because we were really worried about poaching, and we thought these protected areas would be the best place to put them. But we've actually since learned that lobster populations are much too healthy in marine protected areas, and so we're actually doing some of this outplanting in places where there are active lobster fisheries, because that actually helps us remove there's one fewer predator that will chomp on these animals.
Dave Bader comments that there is a white abalone music video coming soon from the Aquarium of the Pacific.
That's fantastic. I can't wait to see it. I will share it heavily on all of the white abalone accounts.
Yeah. Thanks, Dave. And then he says, sweet. Slady has another question. Are there any sustainable abalone farms in Vancouver, British Columbia? And are the abalone sold in supermarkets from these sustainable farms?
So I don't know of any. There used to be one farm near Bamfield, maybe. I don't know of any that are currently operating up there. I know, so the Cultured Abalone Farm in California, Monterey Abalone Company, and then the American Abalone Farm are two of the biggest in the United States. I don't know of any in Canada, and partly because the only species that goes that far north is the pinto abalone, and it's pretty slow growing. So usually when people are doing aquaculture for abalone in North America, they do aquaculture of the red abalone because it's the fastest growing of all of the species. It's actually the largest abalone species in the world. And for aquaculture growers, for commercial growers, growth is kind of the bottom line for production. And so they're going to pick the fast growers. So I think those commercial abalone from California are sold in North American markets, as well as some of them are shipped overseas. There's a lot of abalone aquaculture and even wild abalone harvest in Australia and New Zealand and South Africa as well. And I don't know as much about the sustainability of those aquaculture operations.
Thank you. Do we have any other questions? I have someone typing.
You're welcome.
I'll give it maybe one more minute, and if no one has any questions, we can end a few minutes before 5 Eastern. Again, we will send out the recording to everyone who was on this webinar today. So feel free to share it with your network.
And the video too when Dave sends it.
Yeah, and we'll share the music video, Dave, once that arrives. Please let us know. There might be one more question. you might see this, but this lady asked, how much funding do we have, and where are the funding coming in from for 2020?
So what funds the core part of this program is NOAA Section 6 money. Section 6 money is money that NOAA gives to states for saving endangered species in the ocean. And so it actually flows through California Department of Fish and Wildlife to UC Davis Bodega Marine Lab. And so we basically, from July of 2019 through July of 2021 or June of 2021, we have, 2022. So for 3 years, we have that core funding from NOAA, which is really great. And that basically keeps our pumps running, keeps the lights on, keeps very minimal staff employed to make this work. And then on top of that, basically, most of us are looking for extramural grants through NOAA or other places. So I have a grant from California Sea Grant. I have one through the Salt and Still Kennedy program at NOAA. And I also have one through the Ocean Protection Council in California. And so we are always looking for extra funds. And we are really keen on collaborating with these types of things because like any kind of endangered species restoration, no one has enough time and resources to do it all. And we have so many questions, as you can tell, about how to save this species. And so I would say if any of you have ideas about ways that we can collaborate to get money or to answer some of these questions, I'm really keen on hearing those and to establish more collaborations.
So I think Dave wants to use some of your metaphors and analogies for his staff at the aquarium to talk about while doing interpretation.
Totally. Dave, do you have my email address? Because I'll type my email address in here. Feel free to email me and I can send them over.
Yeah, we'll be happy to collaborate on that. Yeah, he can, yeah, that works too. And then Emma asks, do the abalone have a reproductive expiration date, and do they ever become less capable of reproducing after a certain age?
Yeah, so we don't know a lot about senescence in abalone. Some people think, oh, at some point they just all stop. And some people think that as they get bigger and bigger, they just produce more and more gametes. And we don't know. We also don't know really like what the cycle is for them. Are they every other year spawners? Are they every year spawners? We think white abalone only spawn once a year, but other species spawn 3 or 4 times a year. So these are really basic questions about their biology that we are just scratching the surface of and we don't have answers to yet. It would be great to have them.
Well, if there are no more questions, we'll wrap up. Thank you all for joining us today, and thank you again, Kristen, for the really great presentation. I agree some of the analogies would be, I really want to like use those if I ever talk about abalone. Yeah, and again, we'll send out the recording, and if you're not on our listserv and want to know more about upcoming webinars, please let me know, and I'd be happy to add you. We're hoping to have another one in March or April, so we'll keep you up to date with when that is and with whom. All right, thanks everyone. Thanks again.
Thank you.
Bye, Kristen. Thank you.